Ebola. What comes to your mind when I say that? Panic, disease, epidemic, maybe even death. Now, what comes to your mind when I say Legionella? Probably nothing, right? But presumably, most of you didn't really know about Ebola until it was too late. To tell you a bit about Legionella, it's a bacterial pathogen that causes pneumonia-like illnesses and even death in people with compromised immune systems. So the young, the old, and the sick. To contract Legionella, one inhales it, at which point it invades white blood cells of the lungs and then uses these cells in order to reproduce itself. You typically find this bacteria in man-made water systems, so things like showers, cooling towers, pipes, jacuzzis, fountains, etc. I mentioned Legionella and Ebola together because they're both sources of infectious disease. And I found that when it comes to infectious diseases, the phrase "an ounce of prevention is worth a pound of cure" comes in really handy. Because it's better to prevent a problem from happening than to try and fix it once it's actually happened, right? And how do you prevent a microbe from becoming a problem in the first place? By finding and detecting it first. And as it stands, current detection strategies for Legionella are woefully inadequate in that they take time, are expensive, require a lot of manual labor, or just aren't specific or sensitive enough. And this is where I come in. In order to improve our ability to find this insidious bug, I am designing a biological DNA probe called an aptamer. An aptamer is far more superior to another biological probe you all may know as antibodies. Antibodies are what current diagnostic tests detect in your system in order to find out if you're infected. For example, you get strep, your body has an immune response, and it produces streptococcus antibodies. An aptamer is superior to an antibody because you don't actually need an immune response to make it, nor do you need a specific condition like a temperature of 37 degrees Celsius. What that just means is it's easier to create against any target, be it a drug, or in this case, a bacteria, and you can actually select for the conditions in which you want this probe to work. My goal is that by making this aptamer, this detection tool, we can significantly improve our ability to find Legionella, a bug that in the past 15 years has quadrupled incidences of diseases associated with it in North America alone. My hope is that with the advent of this tool, it'll help a doctor in a hospital, an administrator in a school, or even a sanitation worker for the government make an informed decision and take the appropriate safety measures when dealing with Legionella, so we don't end up having another Ebola on our hands. Thank you for your time.